Welcome all for this lovely session that we're about to have. So I'm Dr. Kennedy Osuka, lecturer in marine ecology at the University of Liverpool. I'd like to introduce our speaker for today. Louise Allcock is a professor of zoology at the University of Galway, Ireland. She began her career working on octopuses in the Southern Ocean and expanded to the deep sea worldwide. Louise has extensively explored submarine canyon systems of the North Atlantic with remotely operated vehicles, leading many expeditions as chief scientist, and has dived in submersibles in the Indian Ocean too. She is passionate about protecting ocean, and you will get to see that as she takes us into the deep ocean. Welcome, Louise.
Thanks very much, Kennedy. Okay, so I didn't honestly know what to talk about today. The book is that got me this invitation to speak to you all is nearly 300 pages of lovely photos of all sorts of deep sea habitats. And I have three amazing co-authors that I wrote it with. And so it covers so much ground. I was like, what shall I talk about? And in the end, I said, I will do the things I know best. And I am literally just off a ship. So I'm sitting in my kitchen here in Galway in the west of Ireland, and I came off a ship yesterday. I've spent the last 3 weeks in the Northeast Atlantic leading an expedition to the submarine canyons that are just on the edge of Ireland's territorial waters. And I have been here. I love this view of Irish waters, and the little brown mass you can see at the top here is Ireland, and all the yellow stuff here is Ireland's continental shelf, and that's about 200 meters deep. And all the blue stuff, the dark green and the blue stuff, is the deep sea, which is what I'm interested in. And in this picture here, if you look right up at the top, you can see a couple of sea mounts that are in the Rockall Trough. That kind of yellow flat disc over on its own, that's the Rockall Plateau where Rockall is. And then this big basin in the middle here is the Porcupine Sea Bight, which I might mention a little bit later. And then here at the center of the picture are canyon systems, submarine canyon systems. And if you drop a camera to the edge of the submarine canyon system here at the head, what you find is this beautiful environment that I had on my opening slide. And here we're looking at like 700 meters deep, 800 meters deep. And what we have is an amazing reef structure. It's what we call a geogenic reef. It's based on the rock structure behind it. It's not the same as a tropical coral reef, which is a biogenic reef, which is created by the animals. Here, all these animals are clinging to the rock, but you can see that it's so rich. It's half a mile down, and it's incredibly rich. Now, this would be completely dark if we hadn't shone the lights of our remotely operated vehicle on it. But you can see that there are huge bivalves there, the big red flesh of the bivalves. There are oysters in here. There are corals. There are little tubularian hydroids, which I'll mention later. So it's fantastically rich. And of course, the first thing that maybe people who have not done deep sea research ask is, how on earth do you get cameras there? Because divers can dive to about 50 meters safely, and we're almost 20 times that depth there. And so the answer is that we put a remotely operated vehicle over the side. Now, this is video that is speeded up because it's quite a slow operation to put it off the ship. This is our remotely operated vehicle, our ROV, as we abbreviate it, or sometimes ROV. And this is Holland 1, and she's going off the side of the Irish National Research Vessel here, Celtic Explorer. And our ROV, you can perhaps see the little yellow thrusters at the back there. That's so that we can steer it and drive it around. You can see a little white box at the bottom, maybe. That's where we store our samples. And on the front of the ROV, we're looking at the back of it, so you can't see it. We've got some robotic arms so that we can pick things up. And we've also got some great camera systems so we can take photos and video. And if we get those camera systems down to that wall I was showing you, this is what it looks like. And you can see better now the size of those bivalves. And here, the laser lights that you can see, those little laser lights are for us to tell how big things are, and they're 10 centimeters apart. So you can see the bivalves were really huge, the size of my hand. And you can see that this gives a really nice three-dimensional habitat with lots of little creatures living there. And then, of course, we get other things like these big congers coming in to feed there. And we often see the congers hanging out in these overhanging caves. If we get a bit deeper, the habitat changes slightly, but it's still got all this wonderful coral. The dark stuff in the background that's a bit sediment covered is stony coral. And we're starting to get octocorals now. The pink things that are swaying in the current there are beautiful octocorals, and you can see that they're covered in all sorts of other creatures. We get a little bit deeper, and the octocorals change species. So we've got a change in what we find in depth. And remember, this would be dark if we didn't have lights on it. So we have these amazing colors, even though we're so dark. And as we get deeper still, you find the abundance drops off a little bit. There's less food the deeper you get. And the types of corals change. Once we get down to this kind of depth, we're mostly on bamboo corals and black corals. And the reason that we really like these coral habitats is that they provide homes to other organisms. So wherever you get deep sea corals, you get much richer biodiversity than you would elsewhere in the deep ocean. And that's why I love them. And I've got some examples here for you. This is a black coral, and it's big and bushy, but almost always you have these little chirostyrid crustaceans living on there. And they seem to spend their whole life living on the corals. And it's a relationship that's of benefit to both animals, both the coral and the crustacean. Crustacean's getting a home. It's getting food that's trapped on the mucus secreted by the coral, and the coral is getting cleaned all the time by the crustacean. So it's a win-win for both animals. Here's different types of corals here. These are octocorals growing, and you can see each of them has a brittle star wrapped around them. And again, the brittle star is getting a home. It's a great place to live. It's up away from the sediment of the seafloor. And it seems the corals benefit too. It hasn't always been well known what benefits the corals get, but you probably remember the oil spill in the Gulf of Mexico, the Deepwater Horizon spill that was so catastrophic, with so much oil released into the Gulf of Mexico, which is a fantastically beautiful, rich habitat. And the deep sea corals there were very impacted by the oil that sunk to the bottom and smothered them. But what researchers found was that those corals that had these asteroschema stars wrapped around them actually recovered quicker, because the stars, the brittle stars, seem to be helping the coral clean itself up. So they increase the resilience. And probably the corals benefit from this on a daily basis, not when there's oil spills, but when there's sediment being churned around. The brittle stars are really doing a lovely job of keeping them clean. So corals enhance the biodiversity, but even dead corals do that. So this is a dead black coral skeleton. It's a coral that is long gone, and it is completely covered with life. And you can see these brasingid starfish here. You can see anemones, flytrap anemones. You can see brittle stars wrapped around the branches. This lacy looking thing here is actually a type of starfish that has branching arms. They call gorgonocephalus, named after the gorgons with their hair. So huge diversity. We've also got little zoanthids growing all over here, and I'll talk a little bit more about them later as well. So corals are great because diversity of the deep sea. And this is more than I guess more than 1,000 meters depth. So there is life in places that maybe we don't think we're going to see it. And the other great thing about corals is they provide habitat for creatures to lay their eggs. So just a couple of years ago in Irish waters, a fantastic shark nursery was found in the coral Mound area. And sharks, you probably know, lay these eggs, which are common name for them is mermaids' purses. They look like little leather wallets with long tendrils coming off each corner of the rectangle. And those tendrils can wrap themselves around coral branches and hang and get well aerated and away safe from the bottom. And so corals do a fantastic job of doing that. This bubblegum coral here, you can see the brittle star arms are wrapped around it, but this is the egg of a Dumbo octopus. So it's not just sharks that are using the corals for laying eggs. Dumbo octopuses do this. And so this egg is about, I would guess, about 2 centimeters tall, and Dumbo octopuses lay their eggs singly. And there will be like one baby Dumbo octopus developing in this, and they don't have a larval stage. It's going to hatch out as a little miniature perfect Dumbo octopus. And we see them a lot in the canyon systems. And this is just because they're beautiful. I put this slide in for that reason, really. So corals are really important as nursery grounds, both for attaching the eggs, but also they provide this 3-dimensional habitat is a really good place to hide. You can imagine if you were, this is an adult Dumbo octopus here, but if you were a baby Dumbo octopus, you would be very vulnerable to fish predators out in this open ocean. And so the cryptic habitat of the corals makes a nice, safe place for nursery grounds. And we've done a bit of DNA sequencing of eggs that we found on various coral species. And it's becoming increasingly clear that quite a few deep sea fishes lay their eggs on corals. Right, I've shown you coral walls, and I've shown you some individual corals. And what I haven't shown you yet is coral reef, deep water coral reef. And there's hundreds of species of deep water coral, all sorts of different types of corals, but only about 5 of them are actually reef building in the deep sea that build actual biogenic reef, where the coral itself builds the reef. And probably the best known and best studied of these is Lophelia. So this is a bit of Lophelia reef. It's had its name changed recently by scientists. Strictly, we should now call it Desmophyllum pertusum, but everybody talks about it as Lophelia. So this is a stretch of Lophelia reef. This is about, I would guess, about 700 meters deep. Again, it's Irish waters. And Lophelia is this stony coral that you see here. It comes in 2 color morphs, which is quite interesting. You've got the sort of orange browny morph, and then you've got the white morph as well. And these reefs are huge. So Lophelia reef is not continuously, but huge stretches of it from the Barents Sea all the way down to South Africa, and on the west of the Atlantic, you get it from Nova Scotia all the way down to Florida. And a few years ago, an area of reef 85 miles long was found off South Carolina. So these reefs are enormous. And they vary quite a lot in their structure. Actually, you can see the first one that I showed was really, really rich in just Lophelia. But here we're on a coral mound. And what happens here is the Lophelia grows, and then it gets sedimented, infilled. And so then more Lophelia grows on top, and then more sediment infills it. And so gradually this mound grows. And in some of the video here, if I just move this on a little bit, you can see that there's actually less live Lophelia here. There's some live Lophelia, but there's a lot of framework. And on top of this, we've got other octocorals growing here. The purple octocoral clavularia is very prevalent. And in this clip here, a different octocoral, Paramaricia, is prevalent. And we're still trying to understand what's the differences in the environment that would lead one mound to be completely covered in one octocoral species, and a different mound just a few miles away to be completely covered in another. And so we've still got an awful lot to learn about these coral habitats. But one thing that you can see about them is, again, they just increase this biodiversity, which is wonderful. Now, what I keep saying is that I keep talking about octocorals, so I felt that I should say something probably about types of corals. So this is an octocoral, and it's very different from a stony coral. And they're really easy to tell what they are because their name means something. They have got 8 tentacles on every polyp. This is a polyp here. In a stony coral, that polyp would be surrounded by calcium carbonate skeleton. But here you can see that's not the case. Stony corals also, they have loads of tentacles. Octocorals just have 8. You can't always count 8 in every single one of them because you don't have the perfect face on view. But they do. And if you snapped this in half, you would find a little rod inside, which is its skeleton. But not all octocorals have that skeleton. Some of them just have tiny little calcium carbonate plates embedded within the flesh. And I mean really tiny, less than a millimeter, like micron size plates that give them their structure. For example, this is a mushroom coral. And this has no solid internal skeleton. It's just stacked with these tiny, tiny little plates called sclerites. And that gives it its shape and structure. And in fact, if you press on this, it is a little bit squidgy, but it's harder than you would think, which is not surprising when it's got these little calcium carbonate rods embedded in it. And again, you can count the 8 tentacles here with their little pinnacles off them. And if I show you a sea pen, here's a really remarkably beautiful sea pen. Sea pens are, in fact, also octocorals. All sea pens are octocorals. And again, you can count the 8 little tentacles around the polyps. And what these do, these have little stinging cells in them, and they capture their prey, and they move it towards the mouth, which is in the center of the polyp here. And then they digest the nutrient values in their food. And then if they obviously, like everything else, there is waste, and they excrete their waste through their mouth. So if you poop through your mouth, there's a good chance you're a coral. You could also be a flatworm, you could be an anemone, but there's a good chance that you're a coral. Okay, and before I move on from coral types, I just want to show you one more. This is a black coral. It's called a black coral because of its black skeleton. They're actually come in all sorts of beautiful colors, which again is remarkable since there's no light down in the deep sea. But you can see the black skeleton, but each of these is a polyp here. There are polyps along all the branches, but they're so packed close together, it's really hard to see them. And you can see them better on the branches here. And these are different because they only have 6 tentacles on their polyps. So they're really easy to distinguish as well. And the interesting thing about black corals is they can't really withdraw their tentacles. When we saw the bubblegum coral with the cirrate egg on, you might have noticed that it didn't really have its tentacles out. And that's probably because it had been disturbed by the ROV settling beside it and perhaps stirring up a bit of dirt in the water. But black corals can't withdraw their tentacles, so they become really, really vulnerable to anthropogenic impacts in the deep sea because they can't escape things. Now, oh yes, so this is not a coral. You can see it's got lots of tentacles. It's got too many to be a black coral. It's got more than 6. It's got too many to be an octocoral because it's got more than 8. It hasn't got a stony calcium carbonate skeleton around its polyp, so it's not a stony coral. And in fact, it's not a coral at all. It's a zoanthid, which is a type of colonial anemone. So it looks just like an anemone with lots of tentacles around it, but you'll see they're all joined together. It's in a colony rather than single like anemones. And zoanthids are cool because they live absolutely everywhere. They were on that dead coral skeleton I showed you earlier. They can live on sponges like this. So this is zoanthids living on a white glass sponge, also in the deep sea. I guess this is about 1,500 meters depth. And they can even form the skeleton or the carapace of a crab. And I really mean this. Well, I'm going to show you. So this is a hermit crab. This is a shallow water hermit crab. Okay, and when you think of a crab, you think of something with 10 legs sticking out and a nice hard shell on the top. This kind of crab doesn't have a hard shell. It has a soft coiled body, which it slides into a borrowed shell that it borrows from a mollusk. So this is a dead snail, and it's nicked the shell. And as this crab grows, it takes a bigger shell from another mollusk. And sometimes these hermit crabs will fight over the best shell, and one will have to give the other up. Now, we get these kinds of hermit crabs in the deep sea. We get all sorts of crabs in the deep sea. So we get hermit crabs like this. We get carrier crabs. See this little carrier crab, he's got his 5th arms up here, and he's holding a bit of black coral as probably camouflage. We don't know exactly why he's holding it, but possibly camouflage. But we also get hermit crabs that instead of having a shell, have a zoanthid, this colonial anemone, instead of a shell. There is no crab carapace under here. There is no shell. This zoanthid is not growing on a mollusk shell that the crab has sequestered. This anemone is forming the shell of this crab. And I got a little video here. They're hilarious. They honestly remind me of Roombas. I just speed this on a little bit as they move along the seafloor. And I think I may have made this twice speed, just to get a little bit more movement in. But at sort of 10 times speed, it's hilarious. It needs Benny Hill music, to be honest. They're such sweet little things. And sometimes we see quite a lot of them. And I suspect that they feed on that they're attracted by perhaps carcasses, that they're attracted to the smell of. Okay. So, I mean, this is just one of the many weird and wonderful things of the deep sea. I think the thing is that deep sea animals are super well adapted and they fill just about every niche that is available. And I'm quite interested in evolution, so I'm really interested in adaptations. So I wanted to show you another very common deep sea adaptation here. I know you're going to get sidetracked by the shark, so I'm going to show you a ghost shark in a minute. But this is a bamboo coral. It's probably nearly 2 meters high. It's absolutely huge. And they have beautiful skeletons. And we're sampling one here for genetics. And I just wanted to show you the cutters that we've developed so that we can sample without damage. Because obviously if something's 2 meters high, you don't want to collect the whole thing. It wouldn't anyway fit in our bio boxes. So we've got a specially designed cutter for the end of our arm there, which cuts and holds. And if we bring that piece into the lab, which is what we've done here, and put it in the dark and just touch it with a pair of forceps, then what you get is bioluminescence. And this is a really common adaptation in the deep sea. Many, many things bioluminesce. You might say, why does a coral bioluminesce? And we think probably that it's kind of like a burglar alarm. So if something else comes along and starts munching on the coral, it grows brightly, and that can attract a predator, a higher predator, to the thing that's munching on the coral. So it's kind of like a defense mechanism. And there's bioluminescence is very common in the deep sea, but it's used for very different purposes. Things like angler fish that you may recall from Finding Nemo, they have this lure that lights up and dangles in front of them, and then something's attracted to the lure, and then they close their mouths around it, and that's how they feed. And some animals, particularly in the twilight zone, in that sort of 400 to 600 meters deep, where there's just tiny bits of light penetrating, they often have little light organs on their bellies so that predators looking upwards can't see them as well, because the twinkly light from their undersides matches the twinkly moonlight on the sea surface. So lots of different reasons for bioluminescence. But I know that some of you found the ghost shark more exciting than the coral. So I've got another picture of ghost shark here. Ghost sharks are amazing. They separated from true sharks about 400 million years ago. And they were really widespread till about the Jurassic, and about 150 million years ago, they were sort of outcompeted by other things. And so they moved down into the deep sea. They're wonderful little things. Some of them have a very pointed nose, and they use that for electroception, for finding their prey with electric signals. And they have all sorts of names. Sometimes they're called rabbit fish, sometimes spook fish, rat fish because of their long tail. But they're really beautiful creatures, and they're very common, particularly in our canyon systems. And just after this clip, I've got a tiny little clip of a baby, which is just so incredibly cute. This is the smallest one I've ever seen, and I put him in for cuteness factor. And there he is. He sees the rov and he just panics and flaps away. And there he was, gone. Right, I'm going to skip that slide because I'm time-wise on. I want to talk, I'm just going to keep talking about amazing adaptations. So this is a sponge. I mean, this was a sponge too, but this is a sponge, and it doesn't have holes in it. Normally a sponge has holes in it, and it draws water through and up out the top, and that's how it feeds. It's filter feeding. It's got little flagellae in there that pull organisms out the water. And you know sponges have holes. You know what a sponge is. You squeeze it and the water comes out. But this sponge doesn't have any holes. What it has is these little fibers instead. And this sponge is carnivorous. And instead of holes and filter feeding, these little spines they're like Velcro, and everything gets stuck to them. And then the cells just wrap themselves around whatever gets stuck to them, and they consume it that way. So we've actually, in the deep sea, we've got carnivorous sponges. And there's about 30 species known now, but they're all really closely related. So it looks like they've just developed once and then radiated a little bit. They're super cool. The other adaptation I want to talk about is giantism, because giantism is really common in the deep sea. On this picture, we go back to this picture, there are these little pink wispy things that are tubularia. They're little solitary hydroids. And this is a deep sea picture. So you get small things in the deep sea, but sometimes you get giant things in the deep sea too. So this is a giant hydroid. And those ones we showed, the polyps on those are about 0.5 centimeter across. But if we look at these are still the laser lights at 10 centimeters. So this hydroid polyp is more than 0.5 meter across. It's just massive, and it's a classic example of deep sea giantism. We see it in more scary looking creatures. This is Bathinomus, which is an isopod. They're very closely related to woodlice, but if I put a woodlouse on that picture at the same scale, there it is. It gives you an idea how big Bathinomus is. I mean, it's more than 1 foot long, and like these things, they have jaws that are amazing, so they're eating machines. And in fact, these have eaten through cables of deep sea scientific experiments. And there's this wonderful quote from Edith Widder, who said, "This is not one of the hazards of deep sea investigations that anyone warns you about." And I just think that quote is lovely. I mean, nobody ever thought to have their experiment eaten by a Bathinomus. And there he is, just very, very calm and still on the bottom. So not very scary, but capable of eating a cable. We see my other example of giantism is in amphipods. So the most common place to see amphipods is on the strand line on the shore. You see these little sandhoppers leaping around, and they're amphipods. This is an example of Antarctic amphipods. They're so diverse, but they're all small. They're all like 0.5 centimeter to maybe 2 centimeters max. This is a deep sea amphipod, and the scale bar down on the right-hand corner is 2 centimeters. So the scale bar would be the normal size of an amphipod. This is a giant amphipod. And that's 20 centimeters long. Imagine if you hold that in your hand, it's like the size of holding a small rabbit. It's absolutely remarkable. But one thing about this, one of the giant amphipod species, not actually this one that's shown here, was actually found with plastic in its gut. It came from an ocean trench like 7 kilometers down. It had plastic in its gut, which is an indication of like how much damage we as humans are doing to the ocean. And we just, I mean, I think everybody listening to this talk probably knows we have to stop getting a plastic in the ocean. It's very difficult to clean it up when it's in there. We have to stop putting it in. Right, but I'm conscious of time. I've been in the submarine canyons. I've been down in ocean trenches looking at ridiculously large things. I just want to talk about chemosynthetic ecosystems for the last 10 minutes of my talk because I love them. I've never worked in them, but I just adore them. They're remarkable. So this video here is from a hydrothermal vent, and you can see the shimmering is from the hot water that comes out of the vent, and it's mineral rich. So it's very rich in hydrogen sulfide. And what happens at hydrothermal vents is in the same way that plants use the energy from sunlight to join carbon together to form sugars, here, bacteria use the energy from the oxidation of reduced compounds, usually hydrogen sulfide. The energy that is released from that chemical reaction is used to combine the carbon together to form sugars. So bacteria chemosynthesize rather than photosynthesize, and we can have really abundant life at these super deep and super extreme environments. So at this particular one, which is in the Western Pacific, we have these hairy gastropods and these alveolid crustaceans. And the shrimps are super cool. They have this little, they don't have eyes, they have these little it is kind of a light organ on their back, a dorsal photoreceptor, and it responds to different wavelengths than visible light, and so it can detect thermal radiation, and this stops them getting too close to the really hot waters. And as you can probably imagine, because very hot water is mixing with cold seawater, the temperature gradient is often quite steep, so your exact position is very important to you to not get fried. And the hairy gastropods, sometimes they just feed on the bacterial mats that are doing the chemosynthesis, but sometimes they actually host these bacteria in their gills so that they have their own little bacteria farm in their gills producing sugars for them. And we get other adaptations at vents. So these are vent barnacles. You might have seen goose barnacles that hang off buoys or the bottoms of ships, and they filter feed. And these are similar to those, but these are remarkably hairy. The hairs on here are super, super fine. And that's so that they can trap bacteria, because bacteria are the base of the ecosystem here. So they're super fine so that they can capture bacteria. And one of the, I guess this is the emblematic vent species that we have here, the giant tube worms. And these are so well adapted. They actually host bacteria within them. Instead of having a gut, they have what's called a trophosome, which is packed with these symbiotic bacteria that are chemosynthesizing. And I always think this is more of an adaptation than having them on your gills, because if you think of something with gills, those gills are open to the environment anyway, because obviously you're ventilating water over your gills to get the oxygen. So it's not quite as internal as having your bacteria in trophosome in the main tube of yourself. And so we have these lovely red plumes sticking out the top. And you know, if you saw them without sort of thinking about it, you might think, oh, this is a feeding plume. But of course, they've got the bacteria to produce their sugars. This is a sort of grabbing oxygen and everything else that the bacteria need plume. And the reason they're so red is they're absolutely packed with hemoglobin to supply that to the bacteria. Which need it for the oxygenation of hydrogen sulfide. So wonderful habitats. And vents aren't the only chemosynthetic habitat. We also have methane seeps. Sometimes they're called cold seeps, just to differentiate them from the heat that you get at hydrothermal vents. But when you look at a cold seep, you see fairly similar sort of organisms. Again, you can see tube worms sticking up here, and you can see that beautiful red color, which shows that they're really oxygen rich and equipped to take oxygen to their symbiotic bacteria. And the symbionts at a methane seep act slightly differently, because we are now talking about methane as the reducing agent, and sometimes the bacteria we have do anaerobic oxygenation of methane, and they produce sulfide as a waste product of that, and then you have other bacteria that then use that sulfide to produce energy. So there's a slightly, perhaps even more complex things going on at the base of the food chain here. And it's not always the same fauna. This is another cold seep, completely different animals. Here we've got large bathymodiolar bivalves. You get these same bivalves at vents. They're slightly different species, but same genus. And again, you see the shrimps as different species than at the hydrothermal vents, but the same kinds of organisms at these cold seeps. And there's two more environments I want to talk about, actually, where chemosynthesis takes place, and this is the first, and this is a whale fall. And whale falls are fascinating, and they're not fantastically known. But if you think about the size of a great whale, this won't happen with a dolphin or a small whale, but with the size of a great whale, think of a grey whale or a blue whale, fin whale. When they die, where do they go? And they go to the deep sea floor. And the first things that arrive are hagfish, and they burrow into them, and the amphipods come because they've got these fantastic mandibles so they can chew through everything. But over time, all the flesh is eaten away. And what happens is bacteria start to degrade the bones, and there's lipid in the bones. And the bacteria use sulfates as a source of oxygen for breaking down the lipids. And the byproduct of this, again, is hydrogen sulfide. So then you get the hydrogen sulfide that you would get at a vent. And so then you start to get, at whale falls, the same kind of fauna that you see at some of the hydrothermal vents. So these are fantastically interesting habitats. We do not know much about them. We know a little bit from people dropping things into the deep sea to study them, but there have not been that many actual found whale falls to date, but there have been some. And they're quite an interesting habitat. But it's really interesting when you think of what we did to the great whales. I mean, we decimated them through whaling in the late 20th century. So this habitat must have been far more prevalent in previous times than it is now with the decreased whale numbers that we have. So that's just something to think about while I show you actually what's going to be, I guess, my last science slide here of what I think is one of the most remarkably strange animals. So this is a worm living on methaneclathrates. So methaneclathrate, it's like methane ice. It's a complex of methane and water molecules that forms into a crystal lattice at pressure. And I mean, I can't think of anything more toxic than a crystal of methane. It can't be good, but for bacteria, it's a source of food. And so bacteria live on this methaneclathrate, and you can see it just looks like ice at the bottom of the deep sea. And these worms, these ice worms, feed on the bacterial mats that grow on the clathrate. So these win my prize for the most remarkable organisms, and to see them in huge abundance. You can see how this is the perfect food source for a particular bacteria, and then that bacteria is in abundance, and so therefore the worms can thrive here. So I'm going to leave you with that slide, but I'm going to give you one thought about climate change to end on, because the deep sea has this methaneclathrate and it's locked up. But if the oceans warm, there's a danger of all this methane being released, and that's just going to accelerate global warming faster. So when scientists talk about tipping points, it's really important that we don't let things go too far, because they can become catastrophic for us if certain things start to break down. And with that, I am going to leave you. I'm going to acknowledge my amazing co-authors, in fact. Hans van Haren is an oceanographer. He's a world-class oceanographer, and he wrote all the oceanography in the book, and he beautifully explains how different water masses affect the life in the deep sea. Monty Preed is the deep sea fish biologist. He's my excuse for not having many fish in my talk, because I don't really need to know about fish when I've got Monty as a co-author. He's absolutely amazing. And Mike Vecchione, if you've ever watched any of the Oceanus Explorer live feeds from the deep sea, you might have heard Mike's voice talking about octopuses and squid. He is a font of knowledge of particularly octopuses and squids, but sort of all things in the deep sea. And I've been in awe of him since I first went to sea with him as a PhD student, and I think I'm probably still in awe of him. And finally, I want to thank people who funded my science, because I wouldn't know anything without them. So I particularly want to thank the Marine Institute, who've provided me with so much ship time. It's their ship, Celtic Explorer, and their ROV Holland 1 that I use. Science Foundation Ireland and the Irish Research Council have funded lots of my research. And the university that I work for, University of Galway, let me go on sabbatical last year, which is what allowed me to write my part of this book. So I'll wrap up there. Thanks so much for listening, and I will be really happy to answer any questions that you have.
Yeah, thank you very much, Louise. That was really thought-provoking. I quite like the ice worms picture, really showing the diversity that's there in the ocean and how different things can utilize different environments. So your talk really has been great, and it has generated some questions. So I have some questions here that have come in. The first question comes from Simon. He asks that, what were you specifically researching on your latest expedition?
Yeah, thanks, Simon. We have built a lot of models of species distributions in the deep sea, firstly. And the reason we have done this is it's incredibly expensive to go to the deep sea, and we cannot look everywhere. So for conservation points of view, it's really important to know what is where, and we can predict this. And what we do is we look at the environment in which these occur, and we make layers of environmental data, and we look at where the species that we're interested in occur, and we look to see where else those environmental data match. We have models that do this for us. Sometimes those models are really high quality, and sometimes they're not, just because we didn't have perhaps the right environmental drivers in there that actually affect the distribution of species. The only way to find out really how good they are is to go and ground truth them. So this particular expedition, we were doing a huge amount of ground truthing, putting the ROV down at test sites where we had a prediction for what we thought was there and seeing if we were right. And we found that some of our models were really good and some weren't so good. And so we'll feed the ones that aren't so good, we can feed new data into them now and try to improve them further. And the ones that are good, we know that they have relevance now to conservation and for putting MPAs in place and that kind of thing.
Yeah, okay, thanks for that. There are also some questions around color. So there's one by Peter. Why in the evolutionary times are there brightly colored corals at depths where it is always dark? And then there's also another one from Helen. Why do the corals in the deep sea have color if it can't be seen with no light there?
Yeah, they're really interesting questions, and they're actually questions that are really difficult to answer. But I will do my best to tell you what we know, which is that so the color is provided by particular chemicals. It's chemicals within the organism that cause light to be absorbed or reflected in this way, and it's likely that those colors are a secondary consequence of other things those chemicals are important for. So a lot of corals, for example, will produce what we call secondary metabolites. They're chemicals that are not crucial for keeping the coral alive, like for its primary life support systems, but maybe they're really important in defense. There's a lot of things growing in the deep sea, and things grow over each other. So some of them will produce noxious chemicals to stop other things growing over them. And possibly it's just a coincidence that some of these chemicals that have this kind of secondary function also happen to absorb a certain light wavelength when you put light on them. So it's probably secondary. In some cases, things do use bioluminescence in a certain way, and certainly some of the reds may be because other creatures with big eyes can see a little bit, but for the most part, it's probably secondary.
Okay, thanks for that. There's also another question still related to color by Anonymous. Are there any reason or theories as to why some corals are so colorful when there is no light in the deep sea? So it seems to be in the same line, but in this case, it's all about theories. Can you shed more insights with theories around the colors of corals?
Yeah, I don't think I can give much further answer to that than I've already given.
Okay. Yeah, so then the other question is on do corals brought up from the great depth then expand as they reach the surface as the air pressure is so much less? So I think these questions around when you bring coral from deeper depth and to the surface, do they expand?
The answer to that is no. So the things that really expand when you bring them up from pressure are air spaces. So anything that has an airspace. So for example, our lungs would be crushed. We couldn't survive. And if you bring something up from deep that has, for example, a swim bladder, then the air in that can expand very quickly, and you'll see that burst. Even from quite shallow depths, you can see that happen. Now, what tends to happen is that things that live very deep don't have air spaces. So they'll have different ways of controlling their buoyancy rather than have air in them. So for example, lots of deep sea squids have have ammonia in their tissues to aid with buoyancy. Others have the chloride ions replaced with sulfates to aid their buoyancy. Things like sharks have very oily livers to help their buoyancy. So the key to being a deep sea species is to get rid of air spaces anyway, because then you don't have problems with pressure. And things like corals don't have any air spaces, so we can bring them up without any problems. Now, sometimes if you take things from really deep, their biochemistry might be particularly adapted to high pressures, and they might not be able to survive when you bring them up because their cellular functions don't work properly at normal pressures, but they won't expand the way that you imagine correctly an airspace would expand.
Okay. Yeah, other question is the warming of the ocean threatening the coral biodiversity in the Irish Sea? If so, how worried are you about the future? And is there anything that can be done?
Yeah, I don't think it is yet, but it will if we don't stop global warming. I mean, one of the things that's going to happen is that current patterns are likely going to change, especially if we have, if ice sheets melt, because the sea ice drives a lot of the global circulation patterns. So if we get changing currents, then we will get different water masses at the depths where we get corals in the Irish waters, and that could be catastrophic. The thing is that like climate has changed in the past, and things have adapted or moved somewhere else, but we're changing the climate so fast there isn't time for adaptation. So it does worry us. I think it probably worries all science, but the only solution, honestly, is to stop burning fossil fuels. And we as a world need to do that. And I don't see a huge move towards that, frankly. And I really, really seriously think we need to.
Yeah. Another question from David is, are estimates available of how much methane gets released at different rising ocean temperatures?
Probably. I don't know what they are. I don't know what temperature rise it would take for the methaneclathrates to start to what's the word? Not to evaporate. Well, anyway, come to the surface and melt. I suppose is the word. So I can't answer you precisely, but I'm quite sure that those data are available.
Okay. Another question from Claire is, I'm hearing that deep sea mining is becoming an increasing threat to the ocean. Can you tell us more about this? I appreciate that it's quite a broad question.
Yeah, it is a threat, and the mining is for the minerals. So we get minerals in various locations. So you get them at hydrothermal vents. They're coming out of the seafloor. We get them actually at seamounts. There's often old manganese-rich crusts at seamounts. We get them on the various places on the abyssal plain, which is the flat, soft ocean, 5,000 meters depth, where you get manganese nodules. The reason that deep sea mining is needed is because we as a world are increasingly wanting these minerals, and particularly rare earth metals, for use in batteries. So things like mobile phones, and also this move to EVs. And of course, there's a sort of governments promoting moves to EVs because they don't want to burn fossil fuels. But actually, the battery requirements of EVs can be destructive to our oceans. So what we really need to do is all move to bicycles and public transport, I'm afraid, because if everybody gets an EV as well as all their mobile phones and tablets, deep sea mining is going to happen. And the problem with deep sea mining is that, I mean, you saw my ROV, it goes down to the bottom, it's tiny, it's got a little robotic arm, it can pick something up delicately. The machines that are going to mine are huge, they've got massive crushing cogs, they churn up everything. They will put loads and loads of clouds of sediment up into the ocean that will take ages to disperse, and that will smother everything. These these habitats are really slow growing. They'll take so long to recover. They'll never recover in our lives, in our lifetime. And of course, as you've seen, they've got really unique animal life that isn't found in other places. So if you wipe it out from there, it's gone forever. And people might say, well, what does it matter? It's at the bottom of the ocean. But actually, we have got enzymes and things that are really super useful from things that live in hydrothermal vents. The PCR testing of COVID relies on bacteria that come from warm environments, hotter environments. So we get all these wonderful things from science and from these extreme environments, and we'll just lose it all. And I really hope it doesn't happen.
Yeah, so still another question from Maggie. Do you have any prediction concerning the evolution of the life forms around the vents?
I wouldn't say really any predictions. Evolution happens so slowly. It happens, along millions of years. If you come back in another 50 million years, I'm sure it will look different, and even more specialized things that you couldn't at this point imagine will have evolved. But I couldn't predict.
Okay. Now there's also another one on how does deep sea life interacts with wrecks? Do they use them like artificial reefs? If so, is there a difference between how they use a wreck versus a rock formation?
Yeah, so deep sea life definitely does settle on wrecks, and they do use them like artificial reefs. What settles there will depend on what the wreck is and what it's made of, what it's painted with. And I wouldn't recommend putting artificial wrecks in place, because you always have unintended consequences. And for example, where you scuttle ships and things, they often have oil associated with them. They're paint. I mean, people paint ships in things that are meant to deter marine organisms for settling on them, because obviously if lots of things settle on the hull of your ship, you get lots of drag and it's difficult. So what settles is very much dependent on what materials the wreck is made out of. But we have seen in plenty of cases, the wreck of the Endurance was found, I think it was just last year, it might have been the year before. Time moves fast sometimes. And there were marine creatures all over it, but perhaps not in such abundance as you might find in a natural environment.
Okay, yeah. So there's also another question on coral bleaching is a big issue in shallow reefs. Does this also occur in the deep ocean under different stress?
So stony corals in shallow waters have symbiotic algae living with them. They're called zooxanthellae, and these algae photosynthesize in the shallow waters using the sunlight that's coming through. And that's one of the reasons why tropical coral reefs are so rich, is because they have a ready-made source of energy there with the symbiotic algae that are part of the stony corals. And in the deep sea, our corals do not have algae because there's no light. So they can be stressed by other things. They can certainly be stressed, but it wouldn't be. And they can certainly be stressed, I'm quite sure, by thermal temperatures. They are used to a very constant temperature environment. That the deep sea it's not uniform, but it's reasonably cold all the time. It doesn't warm up from the sun like the shallow water tropics will. So they can certainly be stressed by changes of temperature, but they don't have any zooxanthellae to lose. So they wouldn't bleach per se, but I'm sure they would be stressed in the same manner. And of course, that is something that could come with global climate change and changing currents. If we get warmer water currents coming over them, then I think it will be very disruptive to our reefs and very destructive, but just not quite in the same way.
So I see we also have some question on Korea, but before we go to those questions, perhaps let's finish with one on biology and all that. So there's one from Christopher. Why do some sea creatures bioluminescence would it be just to attract predators toward them?
Yeah, I totally see your point. I tend to think of everything. I think of everything from an evolutionary point of view, which is not altogether surprising since my background is in evolutionary biology. So for something to be the way it is, it's got to confer an advantage. Otherwise, natural selection wouldn't have selected for it. So I totally get your point that it could attract predators towards them, but it must be working in a way to the animal's advantage. So it must have more benefit in attracting prey to the, let's say, fish than it does in attracting predator. And sometimes it might simply be in the design of those light organs. You know, maybe they're masked from above so that the light is only going downwards, or maybe, you know, masked from a different side so that the light is more available to prey than it is to predators, sort of things like that. But you always have to look at it from an evolutionary point of view, because if it wasn't an advantage, it almost certainly wouldn't be there.
Yeah, so there's also a question about your favorite marine organism and why, and another one on any idea what is the average lifespan of a whale fall ecosystem?
Okay, so let's start with my favorite marine organism. So my background is research on octopuses. I got into the deep sea because I was working on deep sea octopuses. So my favorite organism is a Dumbo type octopus called Storoteuthis satensis, which swims above the seafloor, a little bit like that Dumbo that we saw in the video, but it tends to be further off the seafloor than that. And it has beautiful glowing suckers to attract its prey. And I think it is super cool. So easy answer, Storoteuthis satensis. What was
the average lifespan of a whale fall ecosystem?
Yes, now, difficult one. So whale falls, they go through various stages. The first stage is not a very long one. And that is the hagfish coming in, and the things with really good jaws. Hagfish are cool because they're all slimy and they have these funny cartilaginous keratinized teeth structures, and they bore their way in, and they're all slimy and they can slither in and out of the carcass. That stage isn't very long. And then lots of things come in once the carcass is broken open, and lots of things eat the carcass as quickly as possible. You know, food attracts things from miles around probably. But the slow stage, the long stage, is the degradation of the bones and the release of fat. And there are estimates that that stage can last 50 to 100 years. And some scientists believe that there is a reef stage after that, where the bones just act as hard substrate for things like coral to grow on. And I have seen some images of sponges and coral growing on whale bones, but I've never seen a really rich reef, and I'm not sure that anybody has. So I think that's kind of hypothetical. This could happen, but certainly 50 to 100 years for the bones to break down, for all the lipid to come out of the bones. Yeah.
I think to wrap this up, I'll just pose these questions about the early career. So, what skill would you recommend gaining for an early career researcher wanting to focus on deep sea research? And what motivated you to become a marine scientist?
So I'm going to answer the second one first, and my answer is terrible. Like lots of people have really good career ambitions, and they have plans, and they go and do, you know, what they want to do. And I did very much like octopuses. I was inspired by Jacques Cousteau, who made wonderful programs when I was young. But my real reason for going to do marine biology is that a friend of mine was doing it and loved it, and told me how fantastic it was. And I fell for it. And so I went and tried it too, and I also loved it, and I'm still here. Yeah, that was the first one. What was the first question?
So it's about the career path on becoming an early career scientist.
Oh, yes. get some experience on a ship, I think, because I don't think you can be a deep sea biologist without going to sea. And the first thing you need to find out is it for you? Because it's, 3 weeks on a metal tin box floating on the ocean. If you're one of the people who gets desperately sick, this probably isn't the career for you. Go out and try it and make sure you love being on the ocean. You probably will, but I think that's the first thing to do. And then as soon as you've got some ship time behind you, you've sort of got an open door to help with other expeditions. You've proved your competence. So I would definitely try and get some ship time.
Yeah, with that, we come to the end of this. I first of all want to thank you for agreeing to share this, agreeing to give this talk. It was really a lovely session. We'll look forward to reading more about Deep Ocean. And if you like to purchase a copy of Louise Allcock book, The Deep Ocean, will be available from our partner bookseller, Fox Lane Books. For more information about the sales, please see the festival website or head direct to foxlanebooks.co.uk/festival of ideas. Thank you very much and all the best.